Scientists have reacted to the very intriguing results of a study that identified the drug sildenafil, sold under the brand name Viagra, as a potential candidate for the treatment of Alzheimer's disease, the most common cause of dementia. In the study, led by a team from the Genomic Medicine Institute at the Cleveland Clinic, researchers came to this conclusion after pursuing a multi-pronged approach. First, the researchers identified clusters of genes and proteins that are associated with the mechanisms involved in the progression of Alzheimer's. The disease, which slowly destroys memory and cognitive skills, is thought to be caused by a complex mixture of genetic and environmental factors. Among the main features of Alzheimer's at the biological level are abnormal clumps of misfolded beta-amyloid proteins in the brain, called amyloid plaques, and tangled bundles of another protein known as tau. Another feature is the loss of connection between nerve cells in the brain. After identifying the clusters of genes and proteins, the authors of the study, published in the journal Nature Aging, then used computer modeling to examine how closely more than 1,600 commonly prescribed drugs are known to interact with them, in an attempt to predict which of the substances could. This search narrowed down the list to 66 drugs, of which sildenafil, commonly used to treat erectile dysfunction, as well as high blood pressure in the lungs, appeared to be the most promising candidate when it came to the potential for blocking the damaging processes that occur in Alzheimer's. This analysis found that claimants who had been prescribed the drug appeared to have a 69% reduced risk of Alzheimer's over the following 6 years. This finding remains statistically significant even when accounting for other factors that have an influence on Alzheimer's risk, such as sex, ethnic group, and certain medical conditions. Finally, the authors conducted lab tests to examine how sildenafil interacted with nerve cells derived from Alzheimer's disease patients. They found that the drug reduced certain signs of the disease in the cells. For example, it led to a decrease in tau accumulations. The conclusions of the study support the idea that the drug could potentially treat the disease. Some previous studies in mice and a handful of pilot studies in humans have also indicated that the drug could be beneficial for this purpose. But the authors of the Nature paper stress that its efficacy can only be demonstrated once clinical trials have been conducted that properly assess such a treatment.